Okay, everybody, this is round two of trying to record this the lecture for today. Hopefully, the audio works better this time. So today is our first in a series of lectures about genetics, about identity, about race, and about how we actually make these relationships between identity, race, and our biology. So, what's on the docket for today is we're going to first talk about what ancient DNA is and how archaeologists actually find it. How do they curate it? What do they learn from it? Then we're going to talk a little bit about some recent aDNA discoveries, particularly focusing on Neanderthals and Denisovans. Then we'll conclude the class with a discussion about the particular relationship between Native Americans and aDNA, looking at the Kennewick Man controversy. So let's go ahead and dive in. So in 1984, geneticists recovered 229 base pairs of genetic code from an animal called a quagga, which is basically a subspecies of zebra extinct since the late 1800s. This discovery proved that DNA could survive in dead things and spurred a new field of science called paleogenetics. Today, technological advances in paleogenetics have allowed archaeologists to read the genomes of ancient humans and other organisms, truly transforming our view of history and evolution. So, when organisms die, their DNA slowly begins to decompose. How long this process takes depends on factors like temperature, burial conditions, and the number of microbes decomposing in the body. The general estimate is that in optimal, very, very cold conditions, DNA could survive about 1 million years. To date, the oldest complete genome belongs to a horse, which was unearthed from the frozen ground in the Yukon in northern Canada, and which dates to between about 560,000 to 780,000 years. Most ancient DNA comes from samples younger than 50,000 years old and from very, very cold climates. The oldest DNA from our human ancestors is a fragment of genetic code from a 4,300-year-old Neanderthal, which was found in 2016 in the Cima de los Huesos cave in Spain. Geneticists are starting to have success with aDNA preserved in warmer places, which is particularly important for understanding human evolution in Africa. So the image that you see behind me is a map which shows the location of human remains that have actually yielded whole genomic sequences which paleogeneticists have used to try to recreate genomic sequences in the deep past. So as you'll note here, a lot of those sequences have come particularly from Europe as well as from Western Asia, and only a couple of samples coming from North and Central America, and only one sample in a particularly warm equator climate within Africa. So in the early days of aDNA testing, geneticists could only read these really, really short stretches of ancient genomes. As a result, studies largely focused on tracing either Y chromosomes or what's called mitochondrial DNA, which is linked to maternal lines of ancestry, whereas Y chromosomes, right, are linked to male lines of ancestry. As we'll talk about on Thursday, these really narrow portions of DNA don't actually reflect an individual's complete ancestry, which is actually made up of 23 different chromosome sets. Obtaining DNA from all 23 chromosomes is pretty straightforward for modern populations, but actually poses a lot of problems when we get to ancient DNA. This is because DNA degrades into these tiny little fragments over time that become almost undetectable. And they also undergo chemical reactions that change the code over time, making it really difficult to figure out what the original DNA was connected to. Moreover, aDNA is typically found in these trace amounts within archaeological samples and is often masked by genetic material from all these different microbes that are surrounding the human sample. So what you see in the image behind me are a group of archaeologists taking aDNA samples from a wall within a cave in China in the Gansu province. So one of the major problems that prevented the kind of widespread sequencing of human aDNA for many, many years was this problem of contamination. How do we keep our samples free from all these other radicals that might mess up how we understand the DNA? So genetic material extracted and sequenced from living individuals will consist largely of DNA from that individual, right? In contrast, ancient DNA is so scarce and fragmented that most of the genetic material extracted from fossils tends to be environmental microbes or from the humans who handled those fossils. This human DNA is particularly troublesome since some present day DNA is very similar to that which is sequenced in the ancient DNA samples. So the question is, how do we actually differentiate the modern DNA from the ancient DNA that we're seeing in the archaeological record? Geneticists have developed two key strategies to address this problem of contamination. So there's this kind of standard practice today that you actually only take aDNA samples under these really strict clean room conditions with a filtered air system and UV radiation to minimize the proportion of outside DNA samples that can come in and start to contaminate things. The other big thing that archaeologists and paleogeneticists do is that they add what's called bioinformatic tools to aDNA samples. So bioinformatic tools can be used to remove contaminants or to estimate the proportion of those contaminants present in a DNA sample. So it's basically like a little sticky note that tells us, is this a contaminant which was part of the original sample, or is this good DNA which we can use to actually sequence? So this is a part in the class where I would throw out a question to the group, which is, what can DNA, ancient DNA in particular, tell us about people in the past? So if you're watching this at home or rewatching it for your notes, you can skip over it. If you're watching it at home, make sure to jot down some ideas about how you think aDNA can impact our understanding of the past. For anthropologists, ancient human DNA provides insights that we couldn't learn just from looking at human remains or artifacts. It's already helped provide answers to some major debates, including whether modern humans, Homo sapiens, right, we're Homo sapiens, actually interacted with other types of Homo sapien ancestors, particularly Neanderthals. So despite overlapping ranges of Neanderthals and modern humans in Europe and Western Asia, basically for the last 100,000 years, there's no widely accepted archaeological evidence that they interacted or interbred. So the idea is basically that Neanderthals and Homo sapiens were living kind of side by side, but they weren't necessarily interacting. A 1997 study found that mitochondrial DNA sequences from Neanderthal fossils actually were totally different from the mitochondrial sequences associated from modern humans. This study showed that there was no real admixture between Neanderthal and Homo sapien groups, but it didn't actually provide conclusive evidence against this admixture. Remember, these sample sizes are really small, so even in one given study, we can't make huge generalized claims about all of human history. Nuclear aDNA from Neanderthals helped to resolve this problem, and its analysis showed that actual admixture between Neanderthals and Homo sapiens was occurring in the past. So what archaeologists did was to study 3 individuals who were found in the Vidaja cave in Croatia. These results of aDNA sequencing for Neanderthal genomes were compared to aDNA sequences from individuals located in France, China, Papua New Guinea, as well as from two populations in Africa. The data produced from this comparative study of Neanderthal and Homo sapien genomes found that Neanderthal genomes were more similar to the non-African populations, so those from China and France and Papua New Guinea, than to the two African populations that they sampled. This difference was likely because Neanderthals and the ancestors of present-day non-African populations had interbred in the Middle East and Western Asia, where their ranges were known to have overlapped. Furthermore, comparison of the Vindaya Neanderthal genomes with a larger number of present-day human genomes has revealed the surprising fact that the genetic similarity of Western Asians to Neanderthals is greater than the similarity of Europeans to Neanderthals. So there's a closer connection between modern people living in Western Asia and Neanderthals than there is between modern people living in Europe and Neanderthals. These ancient genomes show that our ancestors not only met, but actually mated with Neanderthals between roughly 40,000 and 100,000 years ago. This intermixing was so common, in fact, that Neanderthal inherited genetic material is found in up to 2% of all non-African populations. So if you're a person out there who has non-African ancestry, the odds that you have trace amounts of Neanderthal DNA in your genomic sequence is pretty high. Ancient DNA research has really kind of complicated this history of interspecies breeding. So in 2010, mitochondrial DNA was extracted from the pinky bone found in this cave called Denisova in Siberia. So when this DNA was sequenced, it was discovered that this little pinky bone represented a sister group of Neanderthals that had diverged from them after the ancestors of modern humans diverged from Neanderthals 500,000 years ago. So we have a divergence between modern Homo sapiens and Neanderthals roughly 500,000 years ago. And then we have a second divergence that happens between Neanderthals and this new group, which came to be known as the Denisovans later on. Scientists refer to this new group as the Denisovans. And these were the first archaic hominin group that was characterized based solely off of genetic material. So previously, archaeologists had used all sorts of morphological features of human bones to try to draw distinctions between species that are part of the human tree. And this species, the Denisovans, were purely based on their genetics, not based on any kind of other morphological characteristics. So genetic research has shown that Denisovans had a different history of admixture than modern humans and Neanderthals. So Denisovan ancestry is found in Melanesians and Aboriginal Australians, and to a much lesser degree in East and Western Asians. This pattern of admixture suggests that Denisovans once had a broader geographic range than Neanderthals, who primarily inhabited Europe and Western Asia. Genetic testing of a little tooth that was found in the same cave in Siberia showed that Denisovans were also more genetically diverse than Neanderthals. Comparisons of Denisovan and Neanderthal genomes led to the conclusion that Denisovans had mixed with other forms of Homo sapien ancestors, so species like Homo erectus and other archaic hominin groups, which diverged from Homo sapiens roughly 1 million years ago. So, in case you're watching this after the fact and you still want to try to get credit for watching the lecture for today, go into D2L and fill out the attendance quiz number 1, which says that the answer to today's quiz is the Pantheon. So make sure you go in and do that. All right, let's keep it moving. So our next section of class, our final section of class, is talking about aDNA and Native peoples. So the first complete genomic sequence from a Native American was published in 2014 by Dr. Rasmussen after testing human remains of a young child identified in Montana. This individual was named Anzic 1 after the young girl who found her. So this individual, Anzic 1, was associated with a large cache of ochre-dusted stone tool technologies. And these technologies are referred to as Clovis points. You can see some of them behind me. These Clovis points date to about 12,600 years ago and are the earliest form of stone technology that we have in the at least in North America. So the Anzic 1 remains were directly ancestral to present-day native populations. And in 2015, Dr. Rasmussen's team conducted genetic tests on another set of human remains that was equally as old. So the remains that they did genetic tests on were called Kennewick Man. And we'll get into what Kennewick Man is all about and some of the controversy around him in a little bit. So aDNA has helped archaeologists answer questions around the timing of human migration into North and South America. In more than a dozen studies, geneticists found that Native people in the Americas stem from 4 major founding maternal haplogroups. Haplogroups is basically just a paleogenetic term for human lineages. So the 4 major groups were A, B, C, D, and then 2 major paternal haplogroups were C and Q. So all of us have these maternal and paternal haplogroups baked into our DNA. The only modern inhabitants of southern Siberia, only the modern inhabitants of southern Siberia match this genetic profile that was found in the haplogroups of native peoples in North America. A large-scale genomic study of 31 present-day humans and 23 ancient human genomes has demonstrated that all native peoples in North America separated from their ancestors in Siberia roughly 20 to 30,000 years ago. Northern Native Americans diverged from their Southern and Central American counterparts roughly 13,000 years ago. This research has shown that there was no single wave of immigration of ancestral indigenous people from Beringia, the ice bridge which connected Siberia to the Americas. Instead, Native Americans received significant recent gene flow through multiple waves of migration from East Asians and Melanesians, especially among South American indigenous populations. Native communities really have a complex relationship when it comes to DNA testing. This relationship has often been one of deep skepticism as a result of unethical practices. So, for example, Native communities provided DNA samples to Arizona State University researchers, particularly blood samples, which were used in a study for diabetes. These samples were then used by researchers at ASU to examine schizophrenia and were transferred to a non-ASU research to study ancient population migrations. So these indigenous samples were used in 2 different studies that were not actually permitted by the original providers of those blood samples. In short, indigenous blood samples serve 3 different research purposes, only one of which is really approved, which really points to the ways in which science has been using indigenous DNA as data rather than as personal property. Another example of this kind of DNA misuse or unethical use came from a study conducted by the University of British Columbia in 2002 with the Nuu tribe in Canada. So what this study did was to take a large number of blood samples during the 1980s and to use those blood samples to study rheumatoid arthritis. The head researcher then took these samples with him when he moved positions to the University of Utah, followed by a new move to Oxford in the UK. And he continued to conduct research on these samples despite taking them to new universities and using them in unrelated research to what had originally been agreed upon. This history of unethical research practices really reflects inequalities and how Indigenous and other people of color are treated by the scientific profession. In addition to these ethical issues, genetic research has been used to challenge Indigenous identity claims and rights over human remains. And this is where the case of Kennewick Man really comes into bear. In July of 1996, two college students stumbled on human remains that had washed up along the Columbia River near Kennewick, Washington. The skeleton, known as Kennewick Man, was about 90% complete and represented a male standing about 5'8" and weighing 170 pounds. Subsequent forensic testing revealed that these bones dated to about 9,000 years old. A storm of controversy erupted when the Army Corps of Engineers, which managed the land where the bones had been found, claimed authority over these remains. A coalition of Columbia River Basin Indian tribes claimed the skeleton under the Native American Graves Protection and Repatriation Act, known as NAGPRA, which was passed in 1990. Under NAGPRA, this coalition of tribes demanded that the bones be handed over for reburial in accordance with their religious beliefs. Basically, the idea that grounded the Indigenous claims for these remains is that the continued scientific study of Kennewick Man was actually a violation of their religious beliefs, which said that human remains should remain in the ground and undisturbed, and that disturbing them would actually have negative impacts on contemporary community members. So as you can see in this quote by Armin Mithorn, who was one of the leaders of this coalition of Indigenous groups calling for the repatriation of Kennewick Man, Native people have a really suspicious relationship when it comes to archaeological research. And we'll get into this in the next couple of lectures, particularly the lecture on biological anthropology. But basically, archaeologists looted fresh native graves, they dug up corpses, and they even decapitated native peoples who had been killed in battle and shipped their bodies to Washington for study. So there's a really complex history of the desecration of Indigenous graves that's part of American history. So in response to the tribe's repatriation request, 8 anthropologists filed a lawsuit to halt the repatriation process. The scientists argued that the remains had to be studied to determine if they actually showed any direct cultural affiliation with modern Indigenous communities. Before repatriation can occur, NAGPRA requires that a tribe must demonstrate a cultural affiliation between human remains and the present-day claimant. Because of the age of the Kennewick Man remains, remember, these are 9,000-year-old remains, the scientists challenged the material and historical evidence that would link these remains to contemporary tribes living in the Columbia River Basin. In 2002, the court confirmed the argument that the bones were not related to any living tribe and that NAGPRA didn't apply. So in class, we talked a little bit about whether folks agreed with the scientist or Indigenous perspective in terms of repatriating the Kennewick remains. Almost 10 years after the skeleton was found, the scientists were given 16 days to examine it. In those 16 days, a team of 22 scientists scrutinized 300 bones and fragments and analyzed those remains to produce a 3D image, which generated this kind of new portrait of what Kennewick Man may have looked like. And it turns out that the portrait that they generated really didn't look like modern native peoples. It had a much narrower skull and a much smaller face on average than native populations today. This led to a series of claims by white nationalist groups that the original Americans were actually of European ancestry. Subsequent studies suggested that the closest living relatives of Kennewick Man were not Europeans, as white nationalists claimed, but rather Maori people from New Zealand, as well as the Ainu people, an indigenous group from Japan. Hoping to find a more definitive type of evidence for Kennewick Man's identity, scientists called for genetic analysis of these bones. So in class, we would talk a little bit about what some of the problems are that might be involved in kind of linking ancient and modern human populations. So one of the big issues here is a numbers problem. So the chance that someone living 9,000 years ago has direct genetic descendants living today would be pretty improbable. If we go back 10 generations, each of us has more than 1,000 ancestors. And Kennewick Man lived over 450 generations ago. So that's 450,000 genetic ancestors that Kennewick Man would have had. Even if an ancient individual did have direct living descendants, they would likely not be found in the strictly maternal or paternal haplogroups, or lineages rather, that can be tested using ancient DNA. A second big issue here is a problem of lineal descent. So lineal descent is a biological concept usually inadequate to represent more inclusive traditional kinship systems that native groups use today. And it's not in line with many tribal membership requirements, which are based on blood quantum rather than lineal descent. Of course, there are some exceptions to this, though. At best, genetic evidence seems unrelated or only complementary to other types of cultural data that can help establish cultural affiliation. Finally, there's the troubling reality of settler colonialism in the Americas, right? So today's ethnic and racial categories, which are so often used as proxies for genetic populations, were not in operation in 1492. It's therefore pretty problematic to use them as reference points for defining relationships to ancient human populations, if they didn't even exist 500 years ago. What would have been the lines of demarcation and the parameters of inclusion for ancient humans? This is a really tough question to definitively answer. Genetic and sociocultural categories are definitely not pinned perfectly together. Even if Kennewick Man had direct genetic descendants living today, such shifts in how identity was categorized would make it really difficult to know whether these ancestors even reside in a population that's part of a federally recognized tribe today. So the big picture here is that genetic evidence rarely gives us conclusive lines of evidence for direct biological relationships. Recent studies have thrown Kennewick Man back into the spotlight. So in 2015, DNA analyses of Kennewick Man remains revealed that his genomes were more closely related to modern Native Americans than any other modern humans in existence. This overturned the previous court's ruling that the remains were not culturally affiliated with Native peoples today, and gave the coalition of Columbia River tribes the right to rebury these remains. So one of the questions that we'll start to get into on Thursday, which I want you guys to start thinking about, is whether genetic definitions of relatedness should really prevail over other types of definitions of identity that we use as contemporary cultural groups. See you guys on Thursday.